Hello and welcome to Nikolay's genetics lessons, and today we are going to talk about gel electrophoresis common mistakes. This one of the more disappointing symptoms you might encounter. Let's say you were extremely meticulous, did everything perfectly and run your gel so that the dye front was perfectly even and right at the bottom. You rinsed the gel in the deionized water, then left it on the shaker. Being late in the day, you forgot to replace the water with a stain. It was stained the next day, but here is the result. So notice that the smaller proteins diffused rather quickly out of the gel, while the larger ones at the top diffused much more slowly and took the stain. The acidified alcohol in the stain solution and the destained solutions is essential. It precipitates proteins, preventing them from diffusing out of the acrylamide matrix. So do not forget to follow the protocol, and such mistakes might lead to what you see on this picture in this gel. So this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.